Imagine you were given a pet alien. How do you care for it? What gases does it breathe? If it breathes. What liquids does it drink? If it drinks. To care for your new friend, you're going to have to figure out its biochemistry. That is, the set of chemical reactions keeping it alive. Or more accurately, is its life? Because life as we know it is just a series of energy harvesting, self-sustaining chemical reactions. When these chemical reactions cease, life becomes death. So how will we figure out what these vital chemical reactions are? How do we figure out an alien biochemistry? Let me show you how. You're watching Bones and Stones. Life is driven by the flow of energy. Things never just become more complex, more ordered, without any help. In fact, the opposite is always happening. That's how the universe is, and nobody really knows why. We just call it the second law of thermodynamics and get on with it. So chemical reactions that build more complex molecules, as in living things, always require the input of energy. Reactions that require the input of energy to make them happen are known as endergonic reactions. To supply this energy, living things will partner endergonic reactions up with reactions that do happen spontaneously, the reactions that release energy, known as exergonic reactions. So life uses exergonic reactions to drive all of the endergonic reactions. What's the best way to do this? Standardise. Imagine a world where you buy a new kettle, but you can't use it anywhere in your house because the plug doesn't fit in any of the electrical sockets. Quite frustrating. That's why all electrical sockets and plug shapes are standardised. We have just one kind of plug socket in our country, so you could just buy any electrical appliance and the plug will always fit. This is very cost efficient, so life on Earth does the same thing. It has one main exergonic reaction that it uses to provide energy for everything else. This key reaction is the breakdown of a molecule called adenosine triphosphate, or ATP for short. Because of this, ATP is often called the energy currency of life. You need to spend different amounts of ATP to get different biochemical reactions going. So you need to make a lot of ATP, approximately your own body weight in ATP every day. In fact, the main reason you eat food, the main reason you breathe air, is just to make more ATP. This process of replenishing your ATP is known as cellular respiration, which is classically defined as the set of chemical reactions that release energy from glucose. So you eat glucose, and the energy from this glucose is released and used to make more ATP. As ATP is the common energy currency, it can then be used to get any other reaction going. A creature's full complement of life-sustaining chemical reactions is known as its metabolism, and each sequence of reactions with an easily definable purpose is known as a metabolic pathway. For example, the metabolic pathway that breaks down glucose into pyruvate, known as glycolysis, is a sequence of 10 chemical reactions. It's the first metabolic pathway in the process of cellular respiration, so it's quite important. Metabolic pathways that break down things are described as catabolic, and pathways that build things are described as anabolic, so glycolysis is an example of a catabolic pathway. If you're ever trying to figure out what an alien eats, then, you need to figure out its key metabolic pathways, the backbone of its metabolism, if you like. For humans, this backbone of metabolism is cellular respiration. It's the set of reactions that explain why we eat sugar, why we breathe in oxygen but breathe out carbon dioxide, and why we drink water. In addition, a lot of other metabolic pathways interlink or overlap with one of the respiration pathways in some way. For example, some of the molecules made in the pathway that follows glycolysis, known as the citric acid cycle, or sometimes Krebs cycle, are diverted onto other pathways which turn them into amino acids, the building blocks of proteins. Yet another reason why you can think of respiration as the backbone of our metabolism. So now we've been through some basic principles, we can now set about investigating our aliens' metabolic pathways. Step 1. Stoichiometry, the posh word for the relationship between the amount of reactants compared to the amount of products at the end of a chemical reaction. So you just stick some alien cells in a tank full of its various gases and food substances, then you measure what goes in and what goes out. Once you've done this, you can write out a balanced reaction equation, which predicts the reactant to product ratio, usually called the stoichiometric ratio. Then you can check how well your experimental results fit with that stoichiometric ratio. A warning though, you better hope you have some clue before you do any of this, because if you just put some random gases in the tank, While you can imagine what would happen if I exposed you to lots of random gases. So hopefully, your alien might have crash landed in a spacesuit full of its atmospheric gases. Maybe with a couple of food sachets in its pockets. Let's try this with an earthbound mystery organism. There's a domain of microorganisms called archaea. They're a bit like bacteria, but they have unique metabolisms not seen in any other life forms on Earth, as you will now see. If you take a colony of a swamp-dwelling species called Methanosarcina barkeri and stick it in a closed container full of its swampy environment, you'll measure 4 changes in the concentrations of gases. Carbon dioxide and hydrogen will decrease, while methane and water will increase. If you try to write this as a balanced chemical reaction equation, you'll get this. This metabolic pathway is called methanogenesis. It's what many archaea have instead of our respiration, and it serves exactly the same function, to release enough energy to make lots of ATP. It can actually metabolize any one carbon molecule, not only carbon dioxide. I've just simplified things for you. However, unlike our complicated multi-pathway respiration, methanogenesis only comprises a sequence of about 9 reactions. So why do we need the more complicated cellular respiration? Why can't we use carbon dioxide instead of oxygen? The answer is that cellular respiration releases much more energy per reaction, because there's much more energy bound up in a single molecule of glucose than in a molecule of carbon dioxide. And as you might expect, huge multicellular organisms like us need much more energy than a few microorganisms to survive. If we relied on methanogenesis, we wouldn't be producing ATP fast enough to last us a second. This leads us nicely onto the next step in investigating an alien biochemistry. Step 2. Bioenergetics, the study of how living things transform energy. As I say a lot, all life is driven by the flow of energy. So you can use this idea to determine which molecules and reactions to focus your efforts on, which are most important energetically speaking. As an example, we can tell that methanogenesis is unlikely ever to sustain any kind of mobile multicellular organism just from our comparison to cellular respiration. Before I can show you how though, I need to explain the basics. How do we measure energy in bioenergetics? Put simply, we use a quantity called Gibbs free energy, named after Josiah Willard Gibbs, who developed the idea in 1878. It's defined as the maximum amount of mechanical work that can be derived from a system under a constant temperature and pressure. I use the word work here, as it's used in physics, to mean the integral of a force exerted on an object with respect to the distance the object is then moved by the force. The change in Gibbs free energy of a chemical reaction, denoted as delta G, is basically an accounting tool that keeps track of 2 key quantities. Delta H, the enthalpy change, i.e. the heat energy given off in the reaction, and delta S, the entropy change from the reaction. Entropy is the amount of disorder in a system. The second law of thermodynamics states that entropy always increases in the universe. Everything is always falling apart into disorder. As I said earlier, this is just how the universe is and we don't know why. Ice always melts when left in the sun. It will never refreeze unless you put in the energy to cool it. So the most important thing to know about interpreting Gibbs free energy is that entropy will only increase like this if the change in Gibbs free energy is negative. This means that a reaction will only happen spontaneously if delta G is negative, in which case the reaction is described as exergonic, like we said before. Conversely, a positive delta G tells you that the reaction is endergonic, and so will not happen spontaneously. In other words, you need to put in energy to get it going. And a delta G of 0 means no net change will occur at all, a state of equilibrium. However, even if a delta G tells you whether a reaction will happen spontaneously, it doesn't tell you how fast the reaction will happen. For example, the decomposition of hydrogen peroxide has a negative delta G, but it happens really very slowly unless you add in a catalyst, defined as a substance that speeds up a reaction without being used up itself. So biological catalysts are very common in living things. We call them enzymes, and you can't have any metabolic pathways without them. On Earth, enzymes tend to be complicated proteins with complicated mechanisms, so alien enzymes will probably be complicated too if they're going to do anything useful. My advice is not to bother worrying about them. The rate of a reaction actually depends on a quantity called the free energy of activation, which is largely unrelated to delta G. So don't worry about the rate of reaction, just focus on the delta G because it's unaffected by enzymes. So back to me showing how rubbish methanogenesis is. Under standard conditions, the delta G of methanogenesis using carbon dioxide is minus 134 kilojoules per mole. whereas the delta G of the oxidative breakdown of glucose is minus 2,870. I say under standard conditions because delta G varies according to temperature and pressure, so keep this in mind if your alien's natural habitat is like a pressure cooker or something. As for Earth's standard biological energy currency, ATP, the breakdown of ATP has a standard delta G of minus 30.5 kilojoules per mole. That's a lot of energy for a small molecule to release, and that's because ATP is a highly unstable molecule due to its high energy phosphorus-oxygen bonds. In fact, the conditions in a living cell increase the delta G for the breakdown of ATP to almost double that at standard conditions, minus 57 kilojoules per mole. You can compare the delta G for different reactions to determine which releases the most usable energy and is therefore likely to be the most important for sustaining life. This is probably the best way to look for whatever your alien uses in place of glucose and ATP. But for a clearer picture, you will of course need to combine delta G with other vital information, such as how commonly any given molecule is found in the universe, which brings us on to our next step. Step 3. Relative frequencies. Top tip: living things evolve to use whatever stuff is most common, because you won't last very long if your survival depends on something very rare. As a good example, the four most abundant elements in the whole universe are hydrogen, helium, oxygen and carbon. If we ignore helium because it's inert, it's then no surprise that hydrogen makes up about 10% of the human body by mass, carbon 19% and oxygen 65%. We are not just any stardust, we are the most common kinds of stardust, and it's reasonable to predict that any alien life will be too. Carbon in particular is not only very common, it's amazing at forming long chains because of its high valency of 4 bonds per atom. Longer chains mean larger, more complex molecules with more complex possible functions, so any alien life not making heavy use of carbon is really missing out. Even many of the more complex molecules used by Earth's living things are common across the universe. For example, astronomers have detected at least one amino acid, glycine, in interstellar dust clouds, and we have many theoretical and experimental reasons to believe that amino acids are being produced across the whole universe, the classic example being the famous Miller-Urey experiments. The upshot of all of this is that some knowledge of Earth biochemistry may be transferable to alien biochemistry. Yes, we might all just use variations on the same theme. If we do, figuring out an alien's biochemistry should be a lot easier. Anyway, you've hopefully now figured out the overarching chemical reactions that form your alien's metabolism, and gained a rough idea of which are most important for keeping the alien alive. So you can now start to go into detail. You can start to break down each main reaction into its constituent sequences of smaller reactions. In other words, you can study the metabolic pathways. In humans, that would mean progressing from knowing about this simple equation you learned in school, to knowing about the individual steps in glycolysis and the citric acid cycle. That's a huge leap in knowledge, particularly useful for doctors and engineers. So how do we make this leap in knowledge? I'm not going to lie, with difficulty. Step 4. Metabolomics. I'll explain, promise. First, you need to describe the organism's full set of metabolites, that is, all of the different types of small molecules floating about in its cells, the reactants, intermediates and products of metabolism. This is known as its metabolome, just like an organism's full set of genes is known as its genome. And just like the study of genomes is called genomics, the study of metabolomes is called metabolomics. There's a whole family of these so-called omic sciences, but I won't go into that here. I just want you to appreciate that before you start working out what reactions are happening, you need to know the full set of things that are available to react and that have reacted. Techniques used in metabolomics, such as liquid chromatography, mass spectrometry, can routinely measure tens to hundreds of metabolites with excellent precision. Once you know what you're working with, the next step would normally be pathway reconstruction, whereby you use computational models to reconstruct metabolic pathways using the metabolite data, pre-existing knowledge about what reactions are known to happen between these metabolites, as well as what metabolic pathways are present in related species of organism. You can even use genomic data alone to predict the metabolic pathways that will be present, just based on the enzymes coded for in said genome. These computational models are the mainstay of this kind of research nowadays, but you just wouldn't be able to use them for a completely alien metabolism. If your alien is full of weird carbonless compounds and strange gases, for example, you just won't have that pre-existing knowledge of what chemical reactions are likely to be happening between any of those exotic compounds. You won't have anything to compare to. So, you'll need to look inside and see what's actually happening. and that's where Fluxomics comes in. Step 5. Fluxomics. Fluxomics is the collective name for the techniques used to determine the flow of metabolites, that is, the rate of turnover through a given metabolic pathway. This is known as metabolic flux. You might now recall that I warned you earlier not to worry about rates of reaction. Well, the word rate is generally reserved for individual reactions, whereas the word flux is used for whole metabolic pathways. Anyway, it's actually useful here. To simplify things greatly, and I do mean greatly, when you know the rates of different reactions, and those reactions have molecules in common, that means the rates will be dependent on each other. By playing with the quantity of reactants, you can then gain an idea as to the likely sequence of these reactions. The main aim though is usually to study how different metabolic pathways are switched on and off by, for example, starvation, temperature changes and other environmental conditions. This is known as metabolic flux analysis. But hang on, you might now think. I just said that in our Alien, we don't know what chemical reactions are happening, let alone metabolic pathways, so how can we possibly measure their metabolic fluxes? Well, with metabolic flux analysis, you can discover new metabolic pathways and calculate their fluxes at the same time. That's the beauty of it. The specific technique I'm referring to is called C13 fluxomics, and the idea is simple. You can trace the pathway of any molecule you're interested in by labelling it with a stable isotope. An isotope, just to remind you, is a variant of an atom that has an unusual number of neutrons and therefore a different mass, and a stable isotope is one that won't radioactively decay over time. C13 is therefore ideal, so you could make a C13 glucose, a glucose which contains a C13 atom. You then bathe some living cells in the C13 glucose and wait for them to do their thing. If you then analyse the resulting metabolites with mass spectroscopy or nuclear magnetic resonance, you'll find that the metabolites of the C13 glucose are all now labelled with C13. Not only does this highlight all of the relevant molecules in the metabolic pathway, but it also gives you an insight into the individual chemical reactions because you can trace the fates of individual C13 atoms by identifying their subsequent locations in each molecule. Yes, you can actually follow that C13 atom along and see what happens to it. But you will then need some knowledge of chemistry to put all the jigsaw pieces together. In addition, you're measuring the concentrations of each metabolite. So once you've worked out those chemical reaction equations with their stoichiometric ratios, you can then infer the patterns of metabolic flux. You know, the whole point of fluxomics. Put all of this atom mapping and stoichiometry together, and you'll be able to build a metabolic network model. That is, a map of the various metabolic pathways and their corresponding fluxes. Just remember that C13 is not your only option for a stable isotopic tracer. Your alien lifeform might not even have any carbon in it to trace. But there's plenty of stable isotopes out there, so just pick one appropriate for your molecule of interest. But wait! What if the alien can change one isotope to another, thereby removing our isotopic tracer from the system altogether? Well, now is a good time to clarify that. As I said from the outset, I'm talking about alien biochemistry. Chemistry, by definition, deals with chemical reactions, which are at the most fundamental level just interactions between electrons. If your alien is able to change one isotope to another, that's not a chemical reaction because it's a neutron interaction. So it wouldn't be chemistry, it'd be nuclear physics. To our knowledge, no life on Earth can transmute one isotope of an element to another. There's no reason an alien lifeform couldn't, except that it would take a hell of a lot of energy. Nuclear fusion powers the sun. Nuclear fission powers our most destructive weaponry and our largest cities. As an aside, I should mention that despite all of this, isotope substitution can change the rate of reactions, even if it doesn't change the reactions themselves. This is known as a kinetic isotope effect, if you want to look it up, but I won't go into detail here. It's just one of the many sources of error to keep in mind when attempting fluxomics. But wait, again! Life need not be based on chemistry. Why couldn't there be a life form composed entirely of matter without any electrons? Or maybe even a being of pure energy, or something else out of a Stephen Baxter novel? Well yeah, feel free to imagine life forms that push our definition of life, but as I said, I was talking about alien biochemistry. So by definition, I was talking about chemistry-based life forms. If I want to speculate about non-chemistry based life forms, I'll do it another time. In fact, maybe I will. I've been talking for long enough. If you enjoyed any of this, like and subscribe because I plan to talk more about alien biochemistry in the future, as well as alien bio-not chemistry. Until then, stay away from venomous snakes.